One bacterium can hand a neighbor resistance to 4 different antibiotics at once in under an hour, without ever dividing. The trick is called bacterial conjugation. On the left is the donor. Inside it floats a plasmid, a small circular loop of DNA, separate from the chromosome, often carrying antibiotic-resistance genes. The donor extends a conjugative pilus, a thin protein bridge that reaches across, grabs the recipient, and reels the 2 cells together into a mating pair. An enzyme called relaxase nicks the plasmid at a precise spot, the origin of transfer, cutting just one of its 2 DNA strands. That single strand is pumped through the bridge, 5' end first, by a molecular machine called a type 4 secretion system. Only one strand crosses. Here is the key. The donor keeps its other strand as a template. Both cells now rebuild the missing complement. This is copy and paste, not cut and paste. Nobody loses their DNA. The recipient now holds a complete copy of the plasmid and instantly becomes a donor itself. 1 cell becomes 2, 2 become 4. Resistance spreads exponentially. Conjugation crosses species lines. A resistance plasmid can jump from harmless gut E. coli into Klebsiella, or Salmonella, which is how the colistin resistance gene MCR, one spread across continents in months. So here's the question. If conjugation only copies DNA and never destroys it, why don't resistance plasmids spread to every bacterium on Earth? What invisible cost keeps them in check when the antibiotics are gone?